Thank you very, very much. Warm good morning, good afternoon, good evening, depending on your location. Thank you to the organizers for giving me the platform to share my thoughts on water microbiome. My name is Augustina Sylverken. I teach at the Kwame Nkrumah University of Science and Technology. I also conduct my research at the Kumasi Center for Collaborative Research in Tropical Medicine. All these institutions are located in Kumasi, all located in Ghana. I joined in the discussions yesterday, and I must say that I learned a lot. But like I said, I would want us to also look at water microbiome. We've been looking at human skin, some infections, and so on and so forth. But for the purposes of my talk, I guess we want to focus on water microbiome. At a point in time, maybe getting to the end of my talk, I will take you, or I will share with you briefly what we have started doing within our institution as far as water microbiome is concerned. So I'm sure we are all very familiar with some of these pictures. Water is all around us. I remember in our usual basic biology class, they will tell us that water is life. And so we all need water. We all may have seen the ocean full of water. We have a lot of water bodies, and importantly, some water even running through the taps in our homes, the taps in our laboratories, and several other places we may find ourselves. But of course, importantly is also some water bodies you find in big cities. Some of them may be polluted, some of them may be clean, and so on and so forth. And it's important that we look at these things. If you decide to visit a water treatment plant, it also has its unique microbiome. We know that typically when you want to look at water microbiome, you want to refer to that entire collection of microorganisms you will find in that water body. But we should also remember that apart from those microorganisms in the water body, there may be some other players or actors in the water body. So you can talk about some other fauna or some flora, and importantly, the physical chemical properties of the water environment itself. In terms of the composition of water, we know that there's a lot of diversity and some dynamism when you want to look at the composition of water. Obviously, sometimes due to even where the water body is located, wherever it is, there may be some environmental stresses on that water body. And at the end of the day, these stresses may also translate into the sorts and the types of microbiome you may even find in that water, especially where you want to compare the microbiome of disinfectant water bodies against non-disinfectant water bodies. In the case of non-disinfectant water bodies, or so-called polluted water bodies or contaminated water bodies, we know that you find a variety of microorganisms, so making up or sometimes even having a change in that composition of its microbiome. So you may find viruses, you may find bacteria, we may find cyanobacteria, we may find algae, we may find fungi, parasites, we may find worms, and so on and so forth. But so, which means that typically in a non-disinfectant water body, chances that you will find all the 5 groups of microorganisms is very possible. Obviously, for such microorganisms, we know that they love to live in that biofilm nature as far as their life is concerned, or how they present themselves or how they live is concerned. So you find all sorts of things, especially in non-disinfectant water bodies. Now, if you even want to look at the composition of water bodies or the microbiome of a water body, which is being taken through the treatment process, so attaining some level of disinfection. Proctor and his friend Hams in 2015 decided to present it so very well. where even in that treatment or that treated or so-called disinfectant or disinfected water body, you will find some groups of microorganisms, so making up that microbiome. However, the good thing over here is that some of these microorganisms may be beneficial. So if, for example, in that water treatment process or plant of yours, you want to employ biological agents in the filtration process, then it means that such microorganisms making up the microbiome of that water body may tend to be beneficial, meaning that at the end of the treatment process, when human beings have consumed the water or they have used it for their daily activities, the expectation is that they don't need to have any potential infection. But obviously, this is debatable. Then at the end of the other stream, is within the same water treatment body, you may even find some detrimental microorganisms, so making up the microbiome of that water body, in this case, a water treatment plant. So these may end up becoming disease-causing agents or pathogens, like we call them, and these can have several effects on the water microbiome, one, and again, on even the end product, which is now the treated water. So things like creating some foul smell in the water, supposed to have been disinfected, things like changing the appearance, the aesthetic value of that water body is also, or that drinking water, or that disinfectant water or disinfected water is also very, very possible. Now, if you have some of these detrimental organisms in the water body or in the water you have treated, then it means that there may be some repercussions, such as some negative effects on human life or on the health of humans who may be consuming the water or using the water for their daily activities. So this is where we may want to have at the back of our minds things like waterborne diseases, water-related diseases, sometimes in situations where the water body itself may create that conducive environment to enable the growth of other microorganisms, or even to enable the growth and the proliferation of other insects, which may also end up transmitting some infections. We know that when it comes to the composition of the water body in terms of its microbiome, we can use it as an indicator for pollution. Now, if we want to use it as an indicator for pollution, then we want to look at that microbiome in two different ways. So we want to look at specific microorganisms, specific microorganisms, and then the levels of those specific microorganisms. So like I said, they may even end up affecting the appearance of the water. Some of the water may look brackish, but they may really not be brackish water bodies because of the presence of some organisms, because the microbial community in that water body may have changed. It may end up affecting the appearance. So the appearance can be an indicator which may draw the minds or the attention of scientists to maybe delve into the possible causes of such a polluted water environment. So the question is, so how do you even want to monitor the water microbiome? Classically, for most microbiologists, when they want to look at water quality and so on and so forth, they may typically use some basic bacteriological methods. So simple plating, culturing, looking out for some total coliforms, some what is known as, or what are known as indicator organisms. So which means that when it comes to drinking water, we need to protect it carefully. We need to monitor it to see whether the microbiome has changed or there has been an increase in the levels of the organisms themselves, so that we can have it as an indicator for pollution. And importantly, it's also necessary that such drinking water is monitored for its quality. But of course, how about viruses? How about parasites? How about worms? How about fungi? Because we know that, look, as for the specific indicator organisms, they may not easily even be identified, but obviously thanks to basic microbiology, basic bacteriological methods, we can use some indicator organisms to see if, or to give, or to provide an indication of the possible presence of these pathogens. But if you want to look out for viruses, you may not be able to use the same methods. Maybe for worms, you may see them physically because they may tend to be macroorganisms. But how about parasites like Giardia? How about fungi, things you may not see readily? As for viruses, now we may even want to monitor the water microbiome for the current coronavirus infection or pathogen. Now the question is, what may be some of the sources of these water contaminants, which may end up changing the microbiome of our water bodies? I think this is very typical for most of us on this call. In our environment, we may tend to see some of these things. There may be some water bodies where you find substances coming from septic tanks easily being released into water bodies. And at the end of the day, they may change the microbiome of the water body, i.e. change even the specific indicator organisms as well as the levels of such indicator organisms. Sometimes human beings can also decide to serve as contaminants. So they may do all sorts of things. They may bath, they may decide to release some fecal matter into the water body. Sometimes you even find spills and runoffs from livestock species. You find cattle running through water, drinking some of the water, direct deposition by humans.
I'm afraid that I'm going to cut you down. So could you please a bit hurry?
But the important thing is it possible for some new pathogens to emerge in such water bodies? Now there's a talk about emerging contaminants. We know that they have a great threat on the water microbiome. They can bioaccumulate, they can magnify, they may even alter some ecological interactions. We know that they can also alter the microbial communities and function. We know that in some water bodies, just the presence of some chemicals may even end up making some organisms in the water body become very susceptible to predations by other organisms. So these are some factors that may cause contaminants to emerge in water bodies, intensive agriculture, like we know, increased urbanization, and of course, climate change. So we have proposed some strategies for mitigation, especially as we inch closely to 2030, where we are all expected to attain sustainable development goals.
I think, thank you so much. I have to cut you out because of the time. So if you have any questions. So there is one question. It says, what are your tools in monitoring the water microbiome? Augustina, can you hear me?
Maybe you can allow her to go to the last slide.
Okay.
So what we have done is to use an existing platform to look at water-based outbreak in peri-urban Africa. And here, what we have done, or what we are doing, is just to sample some water bodies and then perform some sequencing, i.e. go through extraction and sequencing, do some bioinformatics, and then look at some resistance profiling, as well as conduct some in-depth phenotypic analysis of selected isolates. So for us, this is in the initial phases. We started only in December, so I don't have any results to present at this stage. So these are our collaborators helping us to look at water, the content of water microbiome in selected water bodies in Ghana. Thank you.
Sorry to interrupt you. Thank you so much for your nice presentation. I think Alex have a question.
Yeah, thank you very much, Abdi, and thank you, Augustina, for the nice presentation. In fact, I feel very scared when I see the pictures, and of course, reflecting in Africa, where possibly about 1/3 of the population do not have access to quality water. This is very disturbing. And so from your research point of view, Augustina, could you tell us where we are going? Is there something the government is trying to do something about it to improve water quality for the population? Because I'm sure in most parts of Africa, you may have a lot of people consuming water contaminated with fecal contaminants and other things that are very not healthy for the human population in that respect. So maybe, I don't know if you can touch something on that.
Yeah, so I agree with you, Alex. Obviously, when you see such pictures, you need to be scared, especially when you have water running through your taps and you decide to consume such water. We may want to look at policy directions, but obviously, without a baseline research to feed into those policy directions, then we may not really be going anywhere. And once we have collated information, i.e. data, through research, then we can use such data to make some policies, i.e. do we want to indicate that the water quality system within our water treatment system should be really stringent or should be made very stringent, so that even though we may still need some indicator organisms, we may want to change their levels, because we know that the WHO has some levels, but obviously due to sometimes the geographical location of those water bodies, they can end up having differences in terms of the indicator organism. So I will look at policy direction, but before policy direction, I think it will be important for us to collate data. We can decide to do this in a One Health approach, like we have recently adopted to do in Ghana.
Thank you so much. I think due to time compromise, we thank you, Augustina, so much.